Good Monday, everybody. Welcome to week number 5. We are more than halfway through our term now. So this is a good week. We've got a lot going on. You got another milestone due. It's all about DNA, which of course I love, love, love. So let's get to it. Let me get to the next slide. There we go. So last week, what did we learn about? We learned about metabolism in the form of cellular respiration. Most of that respiration takes place in the presence of oxygen, but we have anaerobic respiration and fermentation too for organisms that survive and produce energy without oxygen. You discussed that. Photosynthesis is the process where sunlight is converted into food in the form of glucose by certain organisms on the planet. Everything from cyanobacteria to giant redwoods. So this week, what do we have this week? This week is all about DNA, and I forgot to put that right there. But anyway, it's all about DNA. And so what does the structure of DNA look like? A double helix, right? Wait a minute, get out my favorite prop. Double helix, deoxyribonucleic acid. What does it look like? What it looks like and the structure of it is really important for how it works. So we need to take a look at that. How does DNA replicate? How does it make copies of itself? Well, why is that important? Because when cells divide, you need to have the same DNA in each of the two new daughter cells. So that is replication. Even when organisms reproduce and sexually reproduce, the DNA has to be copied in order to make sex cells. Now, the chromosome number is reduced, but the DNA has to be copied. Also, how does DNA work? So the purpose of DNA is to make all the proteins that the cells need in order to do their biochemistry. Most of those proteins are enzymes, and enzymes run all the chemistry in cells. So that's why DNA is so important, because it contains the genetic information to make all the enzymes that you need to run your chemistry. Okay, the chapters that you need this week are chapters 10, 14, and 15. Please do at least skim through those. And one of them, I can't remember which one now, has the information about cancer cells, and also some about carcinogens. And you will be discussing that. Read your Module 5 overview and watch the videos. However, as I just discovered, the videos are no longer up on YouTube, so I have included video substitutes in the announcement to the right of this video. Okay, over there in the section where I write about what we're doing this week, there I have a link to 3 different videos. One on cell cycle and mitosis, another on replication, DNA replication, and then transcription and translation, how DNA is used to make proteins. Okay, so for your discussion board postings this week, your initial post is all about cancer. So what is cancer? Cancer is cells of yours and mine that divide out of control, and they take over valuable territory in the body. They take up a tremendous amount of nutrition and oxygen from the body. And so they divide out of control, and eventually, if they're not controlled, then your body can't sustain your cancer cells. So the question is cancer a problem with mitosis, which is cell division, and the cell cycle? The cell cycle is also important here, so you need to know about that. Or is it a genetic disease? Is it a disease of genes, of tumor suppressor genes that have been knocked out or damaged, or proto-oncogenes that have turned into oncogenes? So you're going to need to look up information like that. Could it be a disease of the cell cycle where checkpoints are overcome and cells divide when they're not supposed to be dividing because something's wrong with them? So that's the question. Is it one of these or is it both of these? And like I told you before, if anybody ever asks you a question like this and says or both, you can pretty well guess it's both. Because why else would they ask you that? You know, but you're going to have to figure out how to explain that to us. That it might be a problem with the cell cycle, and it might also be a problem with your genes, the particular genes that are present, the little bits of DNA that code for traits. And then in your response post, you're going to talk about environmental carcinogens. Well, what the heck is a carcinogen? A carcinogen is anything that causes mutations in DNA in cells that make those cells become cancer cells. So these are cancer-causing agents. And I know you know of some. So you're going to be discussing two of them and how they might be removed from the environment. We have so many. Just type in carcinogen, list of carcinogens, and the list is humongous. You'll also be working on milestone 3 of the final project. And this is where you get into the last parts of your final project, but you're going to put this information into slides this week. Okay, and this will help you with that final project because you'll have a lot of it already done. So what are the methods of conservation for your particular topic? So if your topic is air pollution, what are ways that we can prevent air pollution? What are some different methods that we can use? Well, we can put catalytic converters on our cars. And okay, I'm not going into too much of it because then I'll tell you. But what are methods of conservation for this particular topic? Why is education important? Is it important in this particular issue? Okay, here's another aside for you. Education is always important on a topic. Why would you just make decisions willy-nilly and not know anything about, oh gosh, wait a minute. Isn't that how a lot of things happen these days? Anyway, education is important. What are the particular solutions that might help this topic? And they may be different from the methods of conservation, these solutions. It depends on your topic. What are the ethical considerations, including is there any legislation that's involved in controlling this particular harmful thing that's going on? What are the ethical components of this? Is there any way to stop it? That sort of thing. What are the positive and negative aspects of the issue? So, for example, deforestation of the rainforest. Is there a negative impact for stopping deforestation? Well, all the native people won't be able to grow crops to make a living. They won't have a place to put their houses, right? So these are negative aspects to this issue. So read through that and make your decisions and do some research on this. And then what's the personal impact that this has on you, and what can you do, you yourself, do to help with this particular problem? Include citations and references. This has been important all along. You have to have 3 citations at least every time you do one of these, and include the references. And like I said before, you need to make these into PowerPoint slides this time. And I cannot emphasize this enough. Label all your sections. How do I know when you're talking about solutions versus positive and negative aspects? Because sometimes people just write on and on and on and on like this is an English essay, and it is not. So label all your sections. That's the only way that I can grade these, because I don't know that you know what the sections are unless they're labeled. Okay? So that's very important. So I'm going to finish up with something that has to do with DNA because it's one of my loves, the use of DNA fingerprinting. So DNA fingerprinting is a way to identify people through the use of DNA. And it is becoming more and more sophisticated absolutely every year. I wanted to give you a little bit of the history so that you get this and just talk about it for a little bit. So DNA fingerprinting was discovered in 1985 by Dr. Alec Jeffries, he has now been knighted, and we call him Sir Alec Jeffries, and here's a picture of him down here. He was and still is from England, and he discovered this technique where he could analyze DNA and tell the difference in DNA samples between people, even between people who are very closely related. And as he was discussing this technique with his wife, she said, that sounds like fingerprinting. So he came up with the term DNA fingerprinting. Now, it doesn't have anything to do with fingerprints. It just means a way to type DNA to identify somebody. This is also called DNA profiling, DNA typing. It's got lots of names to it, but DNA fingerprinting is kind of stuck. And when he discovered this technique, police officers read about this discovery, and they wondered if he could use this to solve some crimes in England. There had been 3 rape and murders in England, and they had not caught the perpetrator yet. And so they asked Dr. Alec Jeffries at that time if he could use this technique to determine who left DNA behind at the scenes of the crime. And he said, yeah, I think I can. Let's give it a go. So what they did was the 3 murders were found in a 3-village area, and they decided to bring in all the men from these 3 villages to take blood samples from them. This was way back when you still had to do blood. They decided to take blood samples from all the men to see if they were a match for the DNA from the 3 crime scenes. Well, during this time, a man, came from one of the villages, and he confessed to the crimes. So the police said, well, this is pretty well solved. He confessed. He gave a lot of detail. And they took some of his blood and sent it to Alec Jeffries. And Alec Jeffries came back to the police, and he said, got good news and bad news for you. They said, yeah? What's the good news? He said, well, I got a good DNA profile for this guy that you sent me a blood sample from. And they said, well, what's the bad news? And he said, doesn't match the crime scene evidence. So they had to keep going and keep collecting blood. Well, as you can imagine, the person who actually did these crimes, the perpetrator, decided that it probably was not a good idea to give his blood. He was thinking, yeah, I think I might get in trouble with this one. So he talked his buddy, Ian Kelly, into giving blood a second time using his name. And his name was Colin Pitchfork, and honest to goodness, I'm not making it up. That's his real name. So he talked his friend into giving blood, which the friend did. But Ian Kelly had the flu, and he went to a pub and started drinking that night. So he wasn't feeling so good anyway, and he was drinking, and his mouth started moving, and he told everybody what he had done. Well, one of the women at the table was very concerned, and she went to the police, and they went to Colin Pitchfork, collected a DNA sample from him, and lo and behold, he was the perpetrator of these 3 crimes. He was convicted, and he will serve life in prison until he passes away in prison. That's the way it works in England. So that was the first case of DNA fingerprinting, and they had collected 5,500 blood samples from the men in those villages, and Alec Jeffries was trying to run all of them. But when they got a suspect and he ran the suspect matched. So this was big news for law enforcement. In 1988, which really isn't that long ago, in 1988, the FBI began using this technique, and that was followed in suit by state police agencies, and now there are local agencies too, especially, in different locations in the country, but I know even around here there are DNA labs. The restriction fragment length polymorphism technique was used first, and that's what gives you the bands like you see on the right-hand side of this slide. This is an old technique now. You need a lot of good, pristine DNA in order to run this technique, and you don't always have that at crime scene, so that was a drawback of it. It does take a while too, and it takes expertise in order to run this technique. Then there was PCR, the polymerase chain reaction, which is used these days to identify cases of COVID. Woo! So you've heard of it before. This is a new technique, the newer technique, and you don't have to have as much DNA. In fact, you can have tiny, tiny, tiny quantities, and it can even be degraded DNA. And there are new technologies being developed. There are technologies where you might be able to test DNA out in the field, which is interesting, but there are all kinds of new advances in DNA as we speak right now. And finally, some of the last things that have been happening with forensic science and DNA is that genealogy websites have been using DNA and DNA profiles to catch criminals. For example, the Golden State Killer, Joseph DeAngelo in California, where he murdered quite a few people and had gotten away with it for about 40 years. But the genealogy website was used to track DNA that was a close match to his, and they finally narrowed it down to him, found his DNA, collected it, ran it, and he was a match for the evidence. He's over 70 now, so he's not going to be in jail for long, but justice will be served in this case because he will go to trial if he's still alive. So that's a little bit about forensic science and DNA fingerprinting and how DNA is used. You will see in the next few years, I will say in the next 10 years, tremendous changes in DNA and brand new technologies that will be faster and more accurate and give us more information. Right now, we can also even take DNA that's been collected and develop a physical profile of the person. So what does this person look like? And they are pretty darn good with these things. Face shape, skin color, of course, hair color, hair texture, height, etc., etc. So we are getting better and better at reading DNA and determining information through someone's DNA. So this is really exciting. So I hope you have a good week. If you're interested at all in forensic science and DNA and forensic science, just let me know. I'm glad to have a discussion with you about it because I sure do love it. And if you have any problems this week, all you need to do is email me, m.sigmon@snhu.edu. I'm here for you. So I hope that you have a wonderful week, and let me know if you run into any problems. All right. Have a good one.